unfortunately, another type of tumor that we frequently see is a very highly malignant brain tumor called a glioblastoma multiforme, a GBM. That tumor is very difficult to treat in the long run. It's oftentimes not very difficult to do surgery on because we can typically remove a very high percentage of the GBM. However, the problem is that up to 2 centimeters away from what you can see in the brain and what you can see on an X-ray, this little tiny microscopic foci of tumor that haven't yet grown. So we can take out everything we see, but there's still GBM lurking that is going to grow again. And so again, we can improve the patient's survival, but in the end, they will unfortunately succumb to the tumor. And it depends on how responsive the tumor is to chemotherapy or radiation as to how long the patient can survive. I've had patients who are very long-term survivors, meaning 5 years, 7 years. Those are outliers almost. Most patients will succumb to the tumor within 2 years, 3 years. It is a little better now than it was 10 to 15 years ago because of certain chemotherapeutic agents that have come out, but it's still a very big problem.
The immune system defends the body against disease. T cells are one of its key soldiers, targeting infected or abnormal cells. But cancer can block those defenses. Now scientists are genetically engineering patients' own T cells to make them smarter and tougher at seeking out and destroying cancer. One version is called CAR T cell therapy. T cells customized to zero in on a patient's specific kind of cancer. It all starts when a patient's blood is filtered through a machine that separates T cells and other white blood cells from the rest. Scientists mix the collected T cells with a virus that has been disabled so it won't cause illness. Instead, that virus carries genetic instructions for the T cells to grow an artificial receptor called a chimeric antigen receptor, or CAR, that will track its cancer target and rev up for an attack. Millions of copies of those engineered cells are grown in a laboratory and given back to the patient intravenously. The first CAR T cells are being tested against types of leukemia and lymphoma that bear a marker, or antigen, named CD19. Once in the bloodstream, those CAR T cells home in on that antigen and grab hold. They release toxic chemicals that trigger a cancer cell's death. As one cancer cell dies, CAR T cells move on to the next, multiplying in the patient's blood until they're no longer needed.
Manufacturing CAR T-cell therapies, the Novartis approach. Novartis has created a sophisticated process for manufacturing chimeric antigen receptor T-cell therapies, or CAR T-cell therapies. Novartis CAR T-cell therapy is developed using the patient's own T-cells, which are part of the body's immune system and play a central role in fighting disease. The first step involves collecting some of a patient's white blood cells, including T cells, through a specialized blood filtration system called leukapheresis. Each patient's cells are assigned a unique identification number, which ensure that the chain of identity is maintained throughout the manufacturing process. The cells are then frozen or cryopreserved and shipped to the Novartis manufacturing facility. After thawing, the T cells are re-engineered when a disabled virus is used to genetically encode the cells. The resulting CAR T cells can now recognize cancer cells and other healthy cells expressing a specific antigen, which are markers on cells that your immune system can identify and produce an immune response against. Next, the modified T cells are multiplied, washed, and undergo a strict quality check prior to release. Then the cells are frozen again and shipped back to the patient's treatment center. The reprogrammed CAR T cells are then infused into the patient's blood, where they have the potential to recognize and fight cancer cells and other healthy cells, expressing the specific encoded antigen or marker.
So what is glioblastoma? It's the most common brain tumor in human beings. And it's one of the failures of oncology since we do not know how to treat it. It is invariably fatal. We know it is resistant to radiotherapy and chemotherapy. And because it's extremely hard to remove it all, because the brain is very packed and it's very intricate, controls the body, controls very sophisticated functions in our body, we use immune therapy because it's very targeted and it uses mechanisms of tumor cell killing that the cells are not necessarily resistant to.
Not only us, but several investigators have explored a different way to harness the immune system, which consists of taking cells from the patient body, making it in the laboratory cancer specific, and then re-infuse them into the patients. So in the end, you re-engineer the patient's immune system to be more powerful and effective.
So in this trial in particular, we take the immune cells from the patient's blood through a simple blood draw. We take them back to the lab in a special facility called the Good Manufacturing Practice Facility. And we genetically engineer them. So we give them a recipe to make a specific molecule that we have designed. And this molecule enables the cell to see some of the tumor characters that it would not otherwise see. We test them in the lab extensively, and once this is ready, we infuse them back into the patient's vein.
We engineer these cells to express what is called the chimeric antigen receptor, which renders these T cells specific for the particular cancer.
So first and foremost, the cells were safe. We did not see any life-threatening side effects. So half of these patients that were eligible had some stabilization of the tumor for a long time, and a number of patients made it past almost to the 3-year mark.
While the results have been encouraging, and we are pursuing now additional studies to make them or improve their efficacy in the laboratory, we are very interested to further engineer these cells. Baylor College of Medicine is one of the few academic institutions in the United States who has a very large good manufacturing facility where we can actually generate these type of products. And that is one of the great strengths, of the college, that we also have a dedicated center, the Center for Cell and Gene Therapies, which enables these type of trials